Hi, everyone. My name is Mortazavi, and I wanted to tell you today about the work that my lab has been doing in conjunction with others in the consortium to characterize the short and long transcriptomes of ENCODE 3 and ENCODE 4. And specifically, I want to tell you today about two very different types of RNA. One is very short, those are MicroRNAs, and the other is are much longer, those are the messenger RNAs. So whereas MicroRNAs are less than 30 base pairs long, messenger RNAs start at 300 base pairs and can go up all the way up to 110,000 nucleotides for the longest transcripts, such as Titin. Fortunately, most of the annotated mRNAs are less than 5 kb long, which means that we can now use modern third-generation sequencing platforms such as PacBio and Nanopore to do full-length transcript isoform sequencing. But we'll return to that in a minute. The first question you might have is, why are we looking at microRNAs as part of ENCODE? Well, whereas most of the consortium has been focused on looking at the function of the chromatin in the nucleus and using mRNA as the output, it turns out that microRNAs are playing an important role as post-transcriptional regulators of messenger RNA. Furthermore, microRNAs start their life typically transcribed as messenger RNA that are processed into precursor microRNAs by drosha, which are further processed by dicer into mature microRNA duplexes. And then one of the strands of this microRNA duplex will end up partnering up with the RISC complex. And depending on how well the sequence is complementary to target messenger RNAs, it will either block translation or cause the degradation of mRNA. Now, for the rest of the microRNA portion of my talk, I'm going to be interested in seeing whether we can detect this degradation of mRNA in a time course of mouse development. So this is work that we published recently as part of the consortium, and this was done in conjunction with Rick Myers' lab, Barbara Wold's lab, and Roderick Legault's lab. And it starts from using the ENCODE 3 mouse developmental time course, where a set of samples were collected, tissue samples were collected across multiple time points of embryonic development, all the way to birth at P0. And we went ahead, and for all of the samples that are colored, we sequenced the microRNAs, and we were able to detect consistent numbers of microRNAs per stage, as well as per tissue. But overall, we detected 785 distinct microRNAs with greater than 2 CPM expression, which means that a lot of the microRNAs we detected in a lot of tissues and a lot of time points. So the next question would be, well, how tissue and stage specific are these? Using a tissue specificity index, we can see that a lot of the microRNAs that we detect actually show relatively little tissue specificity at first, but across development, you get a switch in this tissue specificity so that a substantial number of them show, in fact, quite specific tissue-specific expression by the time that you reach the birth. This is also reflected when you do a global analysis using principal component analysis on all the samples. You can see that very nicely the samples separate by tissue and by time. So principal component 1 separates brain and liver, and you can see that the other tissues are clustering also together. But within brain or within liver, you can see that the earliest stage samples are actually at the bottom, and that over time, the expression increases. And in fact, as you move up PC2, you're also getting further separation on PC1. So this is very intuitively pleasing. This also means that we can go ahead and try to capture that using clustering. So we went ahead and we used massive pro to identify 16 different clusters. And you can see already from the structure of the data that we can see expression levels increasing and that we can get these tissue-specific clusters. Looking at them more closely, we can see, for example, that we recovered multiple brain-specific clusters that differ in terms of the dynamics of their expression. So for example, microRNA cluster 11 are brain microRNAs that are increasing across time, whereas, for example, microRNA cluster 5 are microRNAs that start out high that are going further down. We can not only find brain microRNA clusters, but we can recover clusters specific for the heart, the liver, the lung, and the kidney, and so on. All right, so this is all good, but how does that tie up with the expression of their targets? So here, we decided to use a strategy where we were going to be clustering the microRNA and the mRNAs separately, and then using an ensemble approach to predict the targets of the microRNAs, and then look for an enrichment of microRNA targets in mRNA clusters. So by doing this, we can then look for significant interactions. When we did this analysis, we identified 18 pairs of microRNA-mRNA cluster interactions that were significant, 9 of which had negative correlation between microRNA expression and mRNA targets, which we decided to follow up on. So here's a couple of examples that are representative. So this microRNA cluster 11 that I mentioned earlier, whose expression goes up, we can see that its targets are actually going down in mRNA, right, in the brain. However, the expression of these mRNA targets is much higher in the limb and in the cranioface data. And when we do a Go analysis of these particular genes, we can see that the top Go terms are related to ossification, cardiovascular system development, skeletal system development, blood vessel development, heart development. I.e., we're looking here at a set of genes that are important for the development of these non-brain structures. And these microRNAs are down regulating these substantially. We see a very similar story at the bottom, where we have a cluster of microRNAs that are going higher in the heart, kidney, and lung. And when we look at the expression pattern of their mRNA targets, we can see that, in fact, those messenger RNAs are going down, whereas they're much higher in the brain. And sure enough, when we do a Go analysis of these 450 genes, we can see that these genes are highly enriched for brain development, head development, or brain development. So therefore, we proposed that these clusters of microRNAs that we've identified that are increasing jointly are really there to repress the expression of these developmental genes outside of the tissues where these particular genes should be expressed. Since that study, we've now added a substantial amount of human data sets that are being done in tissue as well as in vitro differentiated cells. And we're going to continue releasing microRNA-seq data sets for ENCODE form. Now, switching over to messenger RNAs and trying to characterize messenger RNA isoforms, if you have a gene where you have a couple of potential distinct alternate splicing events, that would produce, say, only 2 possible isoforms, the one that has all of the exons or the ones that only has the first, middle, and the third exon. Well, if you were going to sequence this by regular Illumina RNA-seq, you are definitely going to be able to detect the splice junctions. However, you do not actually know whether there's an isoform in there that maybe skips the red exon but would incorporate the green exon. This is a very difficult problem to solve with short read sequencing, which is very straightforward to solve with long read RNA sequencing, because what you end up doing there is simply sequencing from the beginning to the end of the read. And the read is your isoform. However, even with this simple scenario, things can get complicated. We have to make some decisions about when we decide that 2 transcripts are the same or they're novel. And we know that we're going to likely observe artifacts related to, say, RNA degradation or, say, incomplete reverse transcription. And we'll have to make decisions about how do we look at an isoform and decide whether it's really novel and how much do we trust it. The last point is that if you're doing a complex project like ENCODE, you would like to be able to track your novel isoforms across time, because ultimately you may actually detect your isoform in one sample, and then you might detect it again in a completely different sample, and you'd like those to have consistent IDs. For ENCODE, we decided to go with PacBio sequencing because we felt that the quality of the reads were higher compared to what we've been able to obtain for Oxford Nanopore. However, ENCODE did this as did some Oxford Nanopore sequencing as a pilot, and I'll briefly mention those results along the way. But we decided that we wanted to build a pipeline that was going to be agnostic, so that it could work equally well from PacBio and from Nanopore. So this particular pipeline, which we call Talon, is already available. And I'm just going to describe some of its major features. So first of all, we start by trying to categorize novelty in a more defined fashion. By that, I mean that given a set of annotated transcripts, we can look at every read, and we need to decide whether the read actually matches a known isoform, even though it may not, for example, get all the way up to the end of the boundaries of that gene, or whether it's actually only matching parts of a known isoform. For example, the intron chain here for this first read matches completely the intron chain for gene 1 isoform A, but here, this particular read only matches from the first two introns, whereas this last one only matches the last one. We categorize these sorts of matches as incomplete splice match. And in our case, we just set these aside. It doesn't mean necessarily that these are not real. For example, I would feel much better if you have cage support or polymerase support. But they need to be considered separately from the other two categories, which we find are more defensible in terms of being true novel isoforms, which are these novel in catalog and novel not in catalog. And the catalog that we're talking about here are splice donors and acceptors. So for example, in the novel in catalog case, what we have is that we have this new exon skipping event that we observe, but otherwise, all of the exons match, the exon boundaries match known exon boundaries. Whereas novel not in catalog, we would actually be observing a novel splice donor or acceptor that has not been seen before. There are additional categories such as genomic and antisense that are useful for categorizing rare events. All right, so as part of ENCODE, we did a pilot study where we first sequenced human GM12878 cells using PacBio cDNA sequencing and 2 biological wraps, and also did direct RNA nanopore sequencing using 2 biological wraps. And then we did a somewhat more interesting study where we compared 2 biological wraps of mouse cortex and mouse hippocampus to each other using only PacBio cDNA sequencing. The talent pipeline actually starts from reads that have been processed and mapped onto the genome using a package such as Minimap. And then those reads are cleaned using another package from my lab called transcript clean, which goes and corrects for small indels and non-canonical splice junctions so that we have the best possible data to feed into talent. And talent itself first starts by looking for internal priming events. We've noticed that a lot of the ISMs that I've mentioned that are prefix ISMs seem to have issues related to internal priming. And so we track that so that we can decide whether to filter based on evidence of internal priming. And then we go ahead and we just annotate each read as being either a known read or a novel read. If it's a known read, we simply assign that read to its isoform and it's left aside alone. But for novel isoforms, we further do a filter based on the idea that the difference between a true isoform and noise is likely to come down to whether we actually detect it more than once. And we detect it in more than one biological replicate. So Talon lets you control those settings so that you can filter the transcripts. And then, for example, in this case, there is, say, a couple of reads from 2 biological reps supporting isoform C, but not isoform D. So therefore, we will actually keep isoform C, but not D. And from there, we can generate 3 different data sets. We can generate a custom transcriptome that shows all of the isoforms that made it through the filtering. And then we can generate this transcript quantification tables, as well as these gene quantification tables. I want to point out that we use all of the reads that map onto a gene for gene quantification, whereas for the isoform quantification, we only track the ones that we could specifically assign to them. And both transcript clean and Talon are available on the Mortazavi lab GitHub. Now, looking first at the GM12878 data, we can see that we have a good gene level reproducibility. We detect most genes that are expressed above 10 ppm, and the majority of genes detected at 1 ppm. 83% of the reads are classified as known, and then another 9% are classified as ISM, and another 6.5% are assigned as being NICR/NMC. And those together account for more than 1,000 models that pass our thresholds and that are detected in GM12878, which has been a heavily studied cell line for the last decade. Doing a transcript level quantification on known isoforms also shows that we have nice reproducibility there, and also that some of these novel isoforms are in fact detected at decent expression levels, suggesting that they are real and that they're consequential. Furthermore, we can go ahead and we can actually score our models using external data sets such as Phantom or, for example, computational poly-A or the ENCODE RNA PET data from ENCODE 2. And you can see that actually our NICs and NNCs score very well using all of these criteria. In fact, better than the known models, which I find remarkable. Now, why does this matter? Because it turns out that if you go to more interesting tissues, such as, for example, cortex and hippocampus, you can see that there are cases where, for example, the major isoform that's detected turns out to be, for example, in hippocampus, one that is known, but that in cortex, it is actually this novel ENCODE isoform, which is the major isoform. And if we didn't know about this novel ENCODE isoform, we would instead redistribute its reads, say, from Illumina sequencing across the other known isoforms, because every one of these splice junctions has been observed before. And that would cause us to essentially misassign the expression level of this read across these other isoforms, which are not as highly expressed. Now, that was a simple example to look at, but if you go to even a moderately complicated locus and recover a lot of new NIC transcripts or NC transcripts, and you try to figure out how to detect genome transcripts, things can become really complicated to do by hand. So my lab has developed actually another package called Swan, which uses graph-based comparisons of the different transcript isoforms in order to try to understand where the differences lie. Novel edges are being shown as these dashed lines, and green represents exons and pink represents introns. Now, Swan started out as a visualization tool, but very quickly we added differential expression test, the gene and transcript level. We now allow for isoform switching detection, as well as novel exon skipping and intron reduction events. And last but not least, we produced these beautiful reports. We did a pilot analysis of this using 5 data sets that were sequenced original on the PacBio SQL1 platform in HepG2 and HFFc6 cell lines. And one interesting example of a novel isoform that we detect is in the ADRM1 gene, where you can see that we actually have this novel exon skipping event where we have an intron that spans several exons that was not in the GenCode annotations. So that's very interesting. And where it really shows why this is useful is that this was an isoform that was flagged as being different, quantitatively different between HepG2 and HFFc6. So whereas the gene is not differentially expressed between the two cell lines, this particular isoform is not detected in HepG2, but is substantially detected in the 3 replicates of HFFc6. Overall, when we did a comparison between those two cell lines, we found 4,000 differential expressed genes and 5,000 differential expressed transcripts, including 279 isoform switching genes, such as ADRM1, where gene expression at the gene level is not changing, but at the isoform level is changing. We also find 285 novel exon skipping events and 47 novel intron retention events. We have now released our first set of 43 PacBio transcriptomes on the ENCODE DCC with Talon as the basis of the ENCODE pipeline, and we are planning on releasing substantially more over the next 12 months. So in summary, our mouse developmental time course of microRNA expression identified microRNA modules that are likely repressing their target mRNAs during development. Talon can reproducibly detect and quantify full-length transcript isoforms. Based on our preliminary analysis, there are still thousands of under-annotated transcript isoforms in human and mouse tissues. And I just showed you that Swan can detect quantitative differences between isoforms in different samples. I wanted to close off by letting you know that ENCODE, in collaboration with GenCode and other members of the long-read sequencing community, is putting up a challenge where we are generating data sets across multiple platforms and across multiple protocols on human and mouse cell types and cell mixtures so that we can test different computational pipelines. The data should be available in November, and we're hoping to take submissions from the community for comparisons in a bake-off. That will run from November to early 2021, with the idea being that we could then analyze this during the first half of 2021. So if you're interested at all, please contact me, or you can find out more information on the GenCode page below. All right, so I wanted to also now just acknowledge the people who actually did the work. So for the MicroRNA, it was primarily Sorena and Ravi who did all of the analysis that I described above. And for Talon, it was Dana, Gabby, and Fairley who did the heavy lifting. And Fairley is also the one who just did Swan. So I want to thank all of our collaborators, Barbara Wolf, Rick Myers, and Roderick Hugo, as well as the whole consortium and NHGRI. Thank you.